The overall goal of this procedure is to illustrate how to track fast-moving organelles, such as lysosomes, moving in 3D within a living cell. This is accomplished by first staining the lysosomes and the microtubules using specific commercial dyes inside cultured living cells. The second step is to use a laser scanning microscope and employ the orbital tracking method to follow the lysosome movement at high speed. The size of the orbit and other parameters such as the frequency response of the system are then selected to perform tracking experiments. Ultimately, the results show that it is possible to follow fast-moving lysosomes in 3 dimensions and to detect interactions with other fluorescently stained organelles encountered along the laser orbit during the tracking.
The main advantages of the 3D tracking method over other existing methods which are based, for example, on astigmatism, is that the 3D orbital tracking method allows us to track over a very large range of the dimension.
This method can help answer key questions in the vesicular transport field, such as how vesicles move along microtubules.
Though this method can be used to study endosomal dynamics, it can also be employed to other systems where it is possible to fluorescently label isolated particles, such as gold nanoparticles, membrane receptors aggregate, nuclear pore complexes, or genetic loci.
Maintain Chinese hamster ovary cells, or CHO-K1 cells, as described